So we're going to be starting on chapter 25, which is the urinary system. So the urinary system, this includes the kidneys, ureters, bladder, and urethra. Now the kidneys do the major work of the system, and the other parts of the system are mainly passageways and storage areas. Now this system maintains homeostasis by managing the volume and the composition or what the solute is made up of the fluid reservoirs and the blood. Here you can see the urinary system in a female. Urine is formed by the kidneys, which are up here. So a kidney here and a kidney here. That urine passes first into the ureters, one on either side, then to the urinary bladder for storage, and then finally through the urethra for elimination from the body. Now there are four major functions of the urinary system. The first one is regarding the kidneys. So they will regulate blood volume and composition. They will also help to regulate blood pressure. So I'll just say regulate blood pressure. Also the blood pH. And glucose levels in the blood. The kidneys also produce two hormones. So these hormones are calcitriol and erythropoietin. We'll talk about those hormones a little bit later in this chapter. And the kidneys also excrete wastes in urine. So I'll fix that there. So excreting wastes in the urine. So that's kind of our first main function, just fully regarding the kidneys. Our second main function has to do with the ureters. So the ureters will transport the urine from the kidneys to the urinary bladder. So it's pretty much transport the urine from kidneys to the bladder. Our third main function is regarding the urinary bladder. So the urinary bladder functions to store urine and expel it to the urethra. And then our final function here, number 4, is regarding the urethra. So the urethra discharges urine from the body. It gets rid of it. So this is the urethra. Now the kidney functions a lot in maintaining homeostasis. So here, these are going to be specific functions or kidney functions that help to maintain homeostasis. So these are homeostatic kidney functions. So the first one here is going to be regulation of the blood ionic composition. So what this means is that the kidneys help to regulate the blood levels of several ions. Most importantly, those ions are going to be sodium, potassium, chloride, and phosphate ions. They also do a little bit of calcium too. The kidneys work to regulate blood pH. So the kidneys will excrete a variable amount of hydrogen ions into the urine. So we're going to get rid of those acidic hydrogen ions, and they'll also work to conserve these bicarbonate ions. And bicarbonate ions are very important, or they are a very important buffer of hydrogen ions in the blood. Now we'll talk about that actually in the next chapter, but these are very important for buffering the pH, for maintaining pH levels in the blood. So both these activities, getting rid of hydrogen ions and conserving bicarbonate ions, will both help to regulate blood pH. Next is the regulation of blood volume. So the kidneys will actually adjust blood volume by either conserving or eliminating water in the urine. So if we're conserving more water, we're holding on to more water, blood volume will increase. But if the kidneys expel or get rid of more water into the urine, then our blood volume will decrease. The next homeostatic kidney function is the regulation of blood pressure. So the kidneys will also help to regulate blood pressure by secreting the enzyme renin. Now renin activates the renin-angiotensin-aldosterone pathway. Aldosterone pathway. So this is a pathway that we will go over later, but just know that this is a very important homeostatic kidney function, as increased renin causes an increase in blood pressure. So we could say increased renin is going to result in increased blood pressure, and we'll go over all the specifics of that later in this chapter as well. Here are some other functions that the kidney has to really help to maintain homeostasis. So the top one here being the maintenance of blood osmolarity. Now by separately regulating the loss of water and the loss of solutes in urine, the kidneys help to maintain a relatively constant blood osmolarity. So those are separate regulations, the regulation of water and the regulation of solutes. That the kidney kind of maintains both of those. Next one is the production of hormones. So we kind of went over this very briefly earlier, but the kidney does produce two hormones, being calcitriol and erythropoietin. Calcitriol is going to be the active form of vitamin D. This calcitriol really helps to regulate calcium homeostasis, and again we'll go over that. And erythropoietin stimulates the production of red blood cells. Red blood cells, RBCs. So erythropoietin is going to stimulate that production. The next homeostatic kidney function is the regulation of blood glucose level. So just like the liver, the kidneys can use the amino acid glutamine in gluconeogenesis. And remember, this forms new glucose molecules. And the kidneys can then release glucose into the blood to help maintain a normal blood glucose levels if blood glucose levels are low. Next is the excretion of metabolic wastes and foreign substances. So by forming urine, the kidneys help to excrete wastes. So these wastes are going to be substances that no longer have a useful function in the body. Now this is an inferior view. So you can see we're kind of like looking up into the body. So we have the left kidney on the right side here and the right kidney here on the left side, just for our view. Now these paired kidneys are reddish kidney bean shaped organs, and they're located just above the waist between the peritoneum and the posterior wall of the abdomen. Now, because their position is posterior to the peritoneum, these organs are said to be retroperitoneal. Now, the right kidney is going to be slightly lower than the left, and this is because the liver really takes up a lot of space on the right side, just superior to the kidney. There is a concave medial border of each kidney that faces the vertebral column. And near the center of the concave border, we have an indentation that we call the renal hilum. Now, the renal hilum, this is where the ureter emerges, along with blood vessels, the renal artery and the renal vein. We also have nerves and lymphatic vessels that emerge from the kidney in this location. Now there are 3 layers of tissue that surround each kidney. The most superficial layer is called the renal fascia. This is a very thin layer of dense irregular connective tissue, and what it really does is it anchors the kidney to the surrounding structures and to the abdominal wall. The middle layer, the adipose capsule, this is a mass of fatty tissue surrounding the renal capsule, which is the next layer, and it also provides the kidney, or protects the kidney from trauma, and it holds it firmly in place within the abdominal cavity. Then we have the deepest layer, which is the renal capsule. This is a smooth, transparent sheet of dense irregular connective tissue, and it's continuous with the outer layer of the ureter. What this renal capsule really does is it serves as a barrier against trauma, and it also helps to maintain the shape of the kidney. Now there are 2 distinct regions inside the kidney. We have a superficial region, which is called the renal cortex. And we'll see these in the next couple pictures to kind of get a visual. And there is a deeper layer called the renal medulla. Now, within the renal medulla, there are several renal pyramids. And the portions of the renal cortex between the renal pyramids, these are called renal columns. Now the renal pyramids consist of the secreting apparatuses and the tubules of the kidneys, and the renal columns will anchor the cortex. So here we can see those different sections of the kidney in a nice little visual. So this is a frontal section through the kidney, and here we can see this superficial light region called the renal cortex. You can see it's a little bit lighter than this darker reddish brown inner region, the renal medulla. In this picture you can also see those several cone-shaped renal pyramids. And we can also see the renal columns between those pyramids. Here we can see a nice detailed anterior view dissection of the right kidney. Here we can see the renal pyramids, and the base of each pyramid, which is going to be the wider end kind of here, this is going to face towards the renal cortex. And the apex of each pyramid, kind of the narrower end down here, This is called the renal papilla, which is labeled there. This points towards the renal hilum. So remember that renal hilum is going to be this little opening, kind of right here. It's going to be labeled this way. Remember, this is where the blood vessels and the ureter all kind of enter there. Now the renal cortex is a smooth textured area that extends from the renal capsule to the bases of the renal pyramids and then also into those spaces between them. We call these spaces the renal column, but those are still parts of the renal cortex. Now together, the renal cortex and the renal pyramids of the renal medulla constitute what we call the parenchyma. I'll do a different color there actually. So parenchyma, this really just means the functional portion of the kidney. So again, the parenchyma is the renal cortex and the renal pyramids. Now within the parenchyma are the functional units of the kidneys. We have about 1 million microscopic structures that are called nephrons. Nephrons are going to be that functional unit of the kidney. And filtrate, which is filtered fluid, maybe that's another term that we should know here. Filtrate. So this is filtered fluid that has come into the kidney, been filtered by the nephrons, and now it's leaving. So this drains into the papillary ducts. So these are going to be here. So here you can see like one little nephron inside the renal cortex and the renal pyramid. Right here, one singular little nephron. Again, there's going to be more than 1 million in each kidney. And so the urine is going to be kind of created here in the nephron. And the filtrate, which leaves the nephron, drains into the large papillary ducts. So that's going to be here at the bottom of the kidney. And these extend through the renal papillae of the pyramids. The papillary ducts will drain into these little cup-like structures that are called minor and major calyces. So first we have a minor calyx. It's going to be this little cup here. And then multiple minor calyx will drain into a single or a couple major calyces here. Each kidney has about 8 to 18 of the minor calyces and about 2 or 3 of the major calyces. The minor calyces will receive urine from the papillary ducts of one renal papilla, and it delivers it to the major calyx. Now, from the major calyces, urine will drain into a single large cavity called the renal pelvis. It's going to be right there. And then from there, urine will drain through the ureter and then to the urinary bladder. So this is just kind of the pathway of how urine is created by the nephron by filtering a lot of fluid. We're going to see that the nephrons will filter blood, and then that filtered fluid will drain through this pathway. And during this pathway, we kind of change the name that we call it. We call it filtrate at the beginning, and then we start to call it urine kind of as it passes down through the calyces. Just kind of changes name. So the kidneys remove wastes from blood, and they also regulate the volume and ionic composition of blood. And because of that, it's really not surprising that the kidneys are really abundantly supplied with blood vessels. Even though the kidneys constitute like less than 0.5% of the total body mass, they receive 20 to 25% of the resting cardiac output by the left and right renal arteries. In adults, the renal blood flow through both the kidneys is about 1,200 milliliters of blood per minute. So it's a very large volume. Many renal nerves originate in the renal ganglion, and then they pass through the renal plexus into the kidneys along with those renal arteries. Remember, those are going to enter by the renal hilum. Most of these are vasomotor nerves that regulate the flow of blood through the kidney by either vasodilation or vasoconstriction of renal arterioles. So really these nerves are really just going to be controlling how much or how little blood flows through the kidneys. So let's talk about the nephron a bit. So like I said very briefly earlier, nephrons are the functional units of the kidneys. Now each nephron consists of two parts. First we have a renal corpuscle. This is where the blood plasma is filtered. And the second part is a renal tubule. And the renal tubule is where the filtered fluid passes. We can kind of see here, just in this picture on the right of a nephron, is that really closely associated with the nephron is its blood supply. And we did just briefly cover the blood supply of the nephron. They do get a lot of blood. Remember, about 20 to 25% of that resting cardiac output is supplied to the kidneys. And so we have these blood vessels you can see all around here, these red arteries all around the tubule. And we will kind of discuss the role of those blood vessels yet. Now, the renal corpuscle itself has two components. These are the glomerulus and the glomerular capsule. This is also called the Bowman's capsule. Now, the glomerulus is a capillary network, whereas the Bowman's capsule is a double-walled epithelial cup that surrounds the glomerular capillaries. Blood plasma is filtered into the glomerular capsule, and then the filtered fluid passes into the renal tubule. Each nephron receives one afferent arteriole, which divides into a tangled ball-shaped capillary network, which itself is called the glomerulus. So this tangled ball that comes from the afferent arterial ball of capillaries is called the glomerulus. Those glomerular capillaries will then reunite to form an efferent arteriole that carries the blood out of the glomerulus. Now, glomerular capillaries are really, really unique capillaries in the body because they're positioned between two arterioles, an afferent and an efferent arteriole, rather than between an arteriole and a venule, as we have seen with other capillary networks. Because they're capillary networks and they also play a really important role in urine formation, the glomeruli are considered part of both the cardiovascular network, or the cardiovascular system, and the urinary system. One really important cell type that I need to mention are the mesangial cells. These are contractile cells, and they help to regulate glomerular filtration. We'll see them in some pictures in a couple slides. Now, the glomerular capsule has visceral and parietal layers. So we're going to have both of these. A visceral layer and a parietal layer. This is just like the serous membranes that we've seen in the past. So the visceral layer here consists of modified simple squamous epithelial cells, which are called podocytes. Now, many foot-like projections of these cells will wrap around the single layer of endothelial cells of the glomerular capillaries, and it forms the inner wall of the capsule. So this is going to be the visceral layers made up of these podocytes that have these little foot-like projections that are called pedicels. Pedicels are like the little feet of the podocytes. And these pedicels will wrap around the glomerular capillaries, and they form the visceral layer of the Bowman's capsule. Now, the parietal layer of the Bowman's capsule, or the glomerular capsule, consists of simple squamous epithelium, and it forms the outer wall of the capsule. Now, filtered fluid from the glomerular capillaries enters the capsular space. This is where that filtered fluid will go into. So this is going to be that space between the two layers of the glomerular capsule. So this is the lumen of the urinary tube. Capsular space, which is the space between the visceral and parietal layers. Think of the relationship between the glomerulus and the glomerular capsule like this. Again, very similar to when we spoke about the serous membranes. You can kind of think of it like the glomerulus is a fist that's punched into a limp balloon, the balloon being the glomerular capsule. And the fist is going to be covered by two layers of the balloon. The layer of the balloon touching the fist is the visceral layer, and the layer not touching the fist is the parietal layer, with a space between them being the inside of the balloon, which we call the capsular space in this case. So just for the sake of this example, we can think of this glomerulus here being the fist here. So that will be the fist, and then this part all around is going to be the limp balloon. All around here. So the fist, again, being the glomerulus, and the balloon being the glomerular capsule. So here we can see the glomerulus, this mass of blood vessels kind of right here. So this is going to be the glomerulus here, and then the glomerular capsule kind of surrounding it totally here. So this being the glomerular capsule here. So here we can also see the mesangial cells. You can see, remember, those mesangial cells are going to be those contractile cells of the glomerulus. We can also see the podocytes here as well with their little feet-like projection pedicels. And remember, these cells are going to be attached to the endothelial cells of the endothelium of the glomerulus. And these mesangial cells and podocytes make up the visceral layer of the Bowman's capsule. In this picture, we can also see the capsular space where that filtrate is going to be filtered into. And we can also kind of see the flow of blood and the flow of the filtrate. So I'll just use a different color here. I'll use a purple so we can see that blood comes in here from the afferent arteriole. Comes into here, it goes into this mass of blood vessels, the glomerulus here. Sorry, not that one. But yeah, gonna come into here. And this is where the cells and everything is going to filter the blood or filter the blood plasma. And whatever is filtered through, whatever gets through the podocytes is going to come out. That was bad color. 1 second, I'll get a different color here. Use darker green. So the filtrate is going to filter out through the cells and everything into the capsular space here. So this is going to be filled with filtrate. Whatever filters through the glomerulus enters into the capsular space. And that filtrate is then going to leave through the proximal convoluted tubule. So this is the renal tubule system, and we'll go over kind of where it goes from there in a later slide. Whatever blood is not filtered kind of stays in the blood vessel. All going to come here, and it's all going to then leave by the efferent arteriole. So that was just a brief overview of the filtration system. We will go into a bit more detail of the glomerulus and glomerular capsule yet. So together, the glomerular capillaries and the podocytes, which completely encircle the capillaries, they form a leaky barrier which is called the filtration membrane. I'll write this down here. The filtration membrane is going to be a combination of the glomerular capillaries and the podocytes. So this is a leaky barrier. So the kind of a sandwich-like assembly of those pedicels from the podocytes allows filtration of water and small solutes, but it does prevent the filtration of most plasma proteins, blood cells, and platelets. So these large proteins and blood cells, they can't actually pass through that filtration membrane, so they stay in the blood. So substances filtered from the blood have to actually cross 3 filtration barriers. So the first one is the glomerular endothelial cells. That's going to be the first barrier of the filtration membrane. Then the second barrier of the filtration membrane is the basal lamina. It's going to be between the endothelium and the podocytes. And then the third barrier is the filtration slit that's formed by the pedicels of the podocytes. So filtration slits. So here we can see those 3 barriers of the filtration membrane. So the first one here is the pores of the glomerular endothelial cells. So the glomerular endothelial cells are going to be very leaky, and this is because of these large fenestrations. Now the large size of the fenestrations allows all solutes in blood plasma to exit the glomerular capillaries, but it does prevent filtration of blood cells and platelets. Now located among the glomerular capillaries and in the cleft between the afferent and efferent arterioles are mesangial cells. Now these are going to be contractile cells that help to regulate glomerular filtration. We'll go a little bit into these mesangial cells a little bit later. The second layer is going to be the basal lamina. It's colored in blue here in this picture. This is going to be a layer of acellular material between the endothelium and the podocytes. So really it just consists of very small collagen fibers and proteoglycans in a glycoprotein matrix. Now there's actually going to be negative charges within this basal lamina matrix that prevent filtration of large negatively charged plasma proteins. Then we have the third barrier here is the slit membrane between pedicels. So here we have a large podocyte surrounding the blood vessel here, surrounding those endothelial cells. Now extending from each of those podocytes are thousands of these little foot-like processes that we call pedicels, and these wrap around the glomerular capillaries. Now the spaces between pedicels are called filtration slits. And there's a very thin membrane, it's called the slit membrane, that extends across each filtration slit. And this allows the passage of molecules having a very small diameter, including water, glucose, vitamins, amino acids, very small plasma proteins, things like ammonia, urea, and some ions to pass through. So only very small molecules are allowed to pass through this slit membrane. Blood plasma is filtered in the glomerular capsule. Remember the Bowman's capsule, which is right here. And then the filtered fluid passes into the renal tubule. So it passes to the left this way. And the renal tubule itself has three main sections. Now, in the order that fluid passes through them, the renal tubule consists of the proximal convoluted tubule. So this is labeled right here. It's going to be that first section that the filtered fluid passes into. So I'll have that right here, number 1. This is also sometimes abbreviated PCT. Then next is the nephron loop. This is also called the loop of Henle. This is here. So this loop of Henle has two sections. We have the descending limb and the ascending limb. So that is going to be, here is the descending limb, and then the ascending limb is at the other end of the loop. And then the third section of the renal tubule is going to be the distal convoluted tubule, labeled here. And this is going to be this little section here, this little wiggly bit. Now just a little bit on what these different terms mean. So proximal, this denotes the part of the tubule that's attached to the glomerular capsule. Remember, proximal means closer to. And distal denotes the part that's further away from the glomerular capsule. The term convoluted, this means that the tubule is tightly coiled rather than straight. One other thing I want to note here too is the locations of different parts of the nephron. So here we can see labeled the renal cortex and the renal medulla with just this little dashed line separating these two sections of the kidney. So we can see that the renal corpuscle and both convoluted tubules lie within the renal cortex. And the nephron loop extends into the renal medulla, makes kind of a hairpin turn, and then returns back to the renal cortex. In each nephron, the final part of the ascending limb of the nephron loop makes contact with the afferent arteriole serving the renal corpuscle. Now, because the columnar tubule cells in this region are really crowded together, they're known as the macula densa. Macula just means spot, and densa means dense. So it's just a dense spot of cells. Now, alongside the macula densa, the wall of the afferent arteriole, and sometimes the efferent arteriole, contains these modified smooth muscle fiber cells that are called juxtaglomerular cells. Now, together with the macula densa, they constitute the juxtaglomerular apparatus. So together, the macula densa plus the juxtaglomerular cells these are going to make up the juxtaglomerular apparatus. This is also abbreviated sometimes JGA. Now, this juxtaglomerular apparatus helps to regulate blood pressure within the kidneys. So here again, we can see the flow of filtrate in the glomerular capsule. In this picture too, we can also see the juxtaglomerular apparatus. So here we have the ascending limb of the nephron loop, where it's really close to the glomerular capsule, where that nephron loop makes contact with the afferent arteriole here. So here we can see these columnar tubule cells in this region that are really crowded together, these macula densa cells. And then we can also see the wall of the afferent arteriole with these modified smooth muscle fiber cells called the juxtaglomerular cells here. So we have this region right here being the juxtaglomerular apparatus. So remember, this is going to be a really important region in helping to regulate blood pressure within the kidneys. So to just briefly go over the flow of filtrate here again, we have blood coming in through the afferent arteriole here, going through the endothelium of the glomerulus, so through here, and then it's going to go through the podocytes of the visceral layer of the glomerular capsule. So remember that we have those podocytes here with their little feet pedicels. And the blood comes into contact with these podocytes and also the mesangial cells, and the blood is filtered here. So the filtrate is going to move through these cells into the capsular space and then out by the proximal convoluted tubule. The blood then is going to leave by the efferent arteriole. So we have filtrate leaving through the proximal convoluted tubule, whatever is filtered through the visceral layer of the glomerular capsule, and then the blood, whatever is not filtered, leaves through the efferent arteriole. Now the distal convoluted tubule begins a short distance past the macula densa. So in this picture here, we can see the macula densa is right here. So the distal convoluted tubule is going to begin just past that on the right side. In the last part of the distal convoluted tubule and continuing into the collecting ducts, there are two different cells that are present. Now these are going to be important cells. Now most of the cells that are going to be within this region, again, this is the last part of the distal convoluted tubule and continuing into the collecting duct. So kind of in about this area here. Kind of going down here, and these are where we're going to find these cells. Now, most cells that we're going to see here are going to be principal cells, and principal cells have receptors for both antidiuretic hormone and for aldosterone. Now, these are two hormones that regulate their functions, regulate the functions of principal cells. Now, there's a smaller number of intercalated cells, and these play a role in the homeostasis of blood pH. Okay, moving on to the two types of nephrons that we have within our kidneys. About 80 to 85% of the nephrons are cortical nephrons. So their renal corpuscles lie in the outer portion of the renal cortex, and they have really short nephron loops, really short loops of Henle. And these nephron loops will lie mainly in the cortex and penetrate only into the outer region of the renal medulla. These short nephron loops receive their blood supply from the peritubular capillaries that arise from efferent arterioles. Cortical nephrons will create urine with osmolarity that's really similar to blood. The other 15 to 20% of the nephrons are juxtamedullary nephrons. I don't know if I discussed this earlier, but the prefix juxta means near to. Anyways, their renal corpuscles lie really deep in the cortex, really close to the medulla, and they have really long nephron loops. Their long nephron loops will extend into the deepest region of the medulla. The long nephron loops will receive their blood supply from peritubular capillaries and also from the vasa recta, and these again are going to come from the efferent arterioles. Something different between the juxtamedullary nephrons and the cortical nephrons is that the juxtamedullary nephrons have thick and thin regions of their ascending limb of the loop of Henle. These nephrons also allow the kidney to secrete very concentrated urine, which is different than the cortical nephrons. Here we can see those two different types of nephrons, the cortical nephron on the left and the juxtamedullary nephron on the right. So here on the left we can see this really relatively short loop of Henle here, whereas on the right, this really extra long loop of Henle going all the way down, almost touching the minor calyx, going into the deepest regions of the renal medulla. Here we can also see the two portions of the loop of Henle in the juxtamedullary nephrons. So here we can see the labeled nephron loop. We have a thin ascending limb and a thick ascending limb. The descending limb is going to be the same all the way through though. Now the lumen of the thin ascending limb is going to be the same as in other areas of the renal tubule. It's only going to be the epithelium that's thinner. Again, remember that nephrons with these really long nephron loops enable the kidneys to excrete very concentrated urine. But it also allows the kidneys to excrete very dilute urine as well. To produce urine, nephrons and collecting ducts perform three basic processes. First is glomerular filtration. Second is tubular reabsorption. And the third is tubular secretion. So first is the glomerular filtration. So this is going to be the first step of urine production. This is going to be step right here, step number 1. This is where water and most of the solutes in blood plasma will move across the glomerular or the wall of the glomerular capillaries, where they are filtered and then move into the glomerular capsule. They are then going to move into the renal tubule. So here we have the renal corpuscle and then the renal tubule and the collecting duct later. So this glomerular filtration is going to be step 1. Next is step 2, which happens right here, which is tubular reabsorption. So as filtered fluid flows through the renal tubules and then through the collecting ducts, the tubule cells will reabsorb about 99% of the filtered water and many of the useful solutes. So we're going to reabsorb that back into the blood from the renal tubule. The water and the solutes are going to return back to the blood as it flows through the peritubular capillaries and the vasa recta. Two terms that we kind of need to know the difference between here. The first one is reabsorption. Term reabsorption refers to the return of substances back to the bloodstream. So essentially you're reabsorbing it back into the body so that it is not excreted in the urine. The term absorption, this is a contrasting term where it means the entry of new substances into the body, like we see in the gastrointestinal tract. So this is going to be entry of new substances. Like in the GI tract. Okay, then our final step or final process in urine production is tubular secretion. This is another term that we should know about as well. So as the filtered fluid flows through the renal tubules and also the collecting ducts, the more material is going to be secreted back into the tubules from the blood. Things like wastes, drugs, excess ions, that kind of thing is going to be secreted from the blood back into the tubule. So here, when we're talking about secretion, this is going to be removing a substance from the blood. And in this case, we're moving from blood into the renal tubule to be excreted. So by reabsorbing and secreting, nephrons will help maintain homeostasis of the blood's volume and composition. So this is going to be a combination of filtration and secretion and then reabsorption. The fluid that enters the capsular space is called the glomerular filtrate. Now, the fraction of blood plasma in the afferent arterioles of the kidneys that becomes glomerular filtrate, this is going to be the filtration fraction. So again, this is the fraction of blood plasma in the afferent arterioles that becomes glomerular filtrate. Now, on average, the daily volume of glomerular filtrate in adults is going to be 150 liters in females and about 180 liters in males. Now, more than 99% of the glomerular filtrate returns to the bloodstream by tubular reabsorption. So that really only about 1 to 2 liters is excreted in urine. The amount of filtrate formed in all renal corpuscles of both kidneys each minute is called the glomerular filtration rate. This is also abbreviated GFR. Now, GFR is directly related to the pressures that determine the net filtration pressure. Net filtration pressure. This is also abbreviated NFP. So the kidneys, any change in the net filtration pressure or the amount of pressure being applied for filtration, this is going to directly affect the glomerular filtration rate. Now, the kidneys do need to maintain a relatively constant GFR. If the filtration rate is too high, then the substances will travel through the glomerulus too quickly, and then nothing can be reabsorbed, or not enough will be reabsorbed. If the GFR is too low, then nothing will be excreted because then everything's going to be reabsorbed. So we have to have that really good balance and have a relatively constant GFR in the kidneys. So the glomerular filtration rate depends on 3 main pressures. One pressure promotes filtration and 2 pressures oppose filtration. So our first pressure we're going to talk about is the glomerular blood hydrostatic pressure, or GBHP. Now, this is the blood pressure within the glomerular capillaries. This pressure promotes filtration, and it promotes filtration by forcing water and solutes in the blood plasma through the filtration membrane. Generally, the GBHP is about 55 millimeters of mercury. The next pressure is the capsular hydrostatic pressure, or CHP. Now, this is the hydrostatic pressure that's exerted against the filtration membrane by fluid that's already in the capsular space and the renal tubule. So it's pressure that's already, or fluid that's already in these spaces. It's kind of pushing back on that filtration membrane. Now CHP opposes filtration, and it kind of represents a back pressure, and this back pressure is about 15 millimeters of mercury. Then the last pressure is the blood colloid osmotic pressure, or BCOP. Now we did see this similar pressure when we looked at the capillary net filtration pressure in the circulatory system, so just kind of think back to that. Now the blood colloid osmotic pressure is due to the presence of proteins like albumin, globulins, and fibrinogens in blood plasma. And this pressure also opposes filtration. Now the average BCOP in glomerular capillaries is about 30 millimeters of mercury. So that, if we kind of add those all together, we get the net filtration pressure. And just very quickly remember that that net filtration pressure is directly related to glomerular filtration rate. So the rate at which the glomerulus filters blood plasma. So we get our net filtration pressure, the total pressure that promotes filtration. This is going to be the equation by adding all these together. So as the GBHP promotes filtration, and the other two oppose filtration, we're going to subtract CHP and BCOP from the GBHP. So we can see here we have 55 minus 15 minus 30. So therefore a pressure of about only 10 millimeters of mercury causes a normal amount of blood plasma to filter from the glomerulus into the capsular space. In adults, the GFR averages about 125 milliliters per minute in males and 105 milliliters per minute in females. Now there are three mechanisms that control GFR. These are renal autoregulation, neural regulation, and hormonal regulation. So renal autoregulation, this is the capability for the kidneys themselves to help to maintain a constant renal blood flow and GFR, despite normal everyday changes in blood pressure, for example, like that happen in exercise. So this is just the ability of the kidneys themselves to help to maintain constant renal blood flow and GFR. Next is the neural regulation. Just like most blood vessels of the body, the blood vessels of the kidneys are supplied by the sympathetic autonomic nervous system fibers that release norepinephrine. Now at rest, the sympathetic stimulation is going to be relatively low, but with some stimulation, like during exercise or even during hemorrhaging, vasoconstriction is going to occur, and blood flow to the glomerular capillaries is going to be greatly decreased. So during some stimulation, that neural regulation is going to cause vasoconstriction of the kidney, or the blood vessels of the kidney, and it's going to decrease blood flow to the glomerular capillaries and decrease that GFR. Then we have hormonal regulation. So there are two hormones that contribute to GFR. These are angiotensin II, and this reduces GFR. And then we have the atrial natriuretic peptide, ANP, and this is going to work to increase GFR. So let's go over those three mechanisms that control GFR in a little bit more detail. So first off, we have the renal autoregulation. So the kidneys themselves, like I said, help to maintain a constant renal blood flow and GFR, despite these normal everyday changes in blood pressure like we have during exercise. So this is called renal autoregulation, and there are two main mechanisms for renal autoregulation. The first is the myogenic mechanism, and then we have the tubuloglomerular feedback. Now, working together, these two mechanisms maintain a nearly constant GFR over a wide range of systemic blood pressures. So the myogenic mechanism occurs when stretching triggers contraction of smooth muscle cells in the walls of the afferent arterioles. So as blood pressure rises, the GFR also rises, and this is because renal blood flow increases. Now, the second contributor to renal autoregulation is the tubuloglomerular feedback, and it's named this because part of the renal tubules, the macula densa, this provides feedback to the glomerulus. Now, when the GFR is above normal due to elevated systemic blood pressure, the filtered fluid flows more rapidly along the renal tubules. Now, as a result of this, the proximal convoluted tubule and nephron loop have less time to reabsorb sodium and chloride and water. So in these cases, the macula densa is going to inhibit the release of nitric oxide. And if we remember, nitric oxide causes vasodilation. So when we're inhibiting the release of nitric oxide, we're inhibiting vasodilation, which causes the afferent arterioles to constrict. And as a result, less blood is going to flow into the glomerular capillaries, and we're going to consequently get a decrease in GFR. So here we can see that negative feedback loop for the tubuloglomerular feedback. So here we get an increase in GFR at the top. And the macula densa cells of the juxtaglomerular apparatus are going to provide this negative feedback regulation of the glomerular filtration rate. So when the filtration rate increases, these macula densa cells are going to detect this. They're going to detect the increased delivery of sodium and chloride and water. When they detect this, they are going to decrease the secretion of nitric oxide. In this case, the control center is the juxtaglomerular apparatus itself. Now, because nitric oxide causes vasodilation, the afferent arterioles are going to constrict when nitric oxide is decreased. And as a result, less blood is going to flow into those glomerular capillaries, and the response is going to be a decrease in GFR. So then we're going to get a return to homeostasis, bring that GFR back to normal. One thing to note is that the tubuloglomerular feedback does operate more slowly than the myogenic mechanism. Like most blood vessels of the body, those of the kidneys are supplied by the sympathetic autonomic nervous system fibers. They release norepinephrine. Now, norepinephrine causes vasoconstriction, and this is through activation of alpha-1 receptors. Don't really have to memorize this, but the alpha-1 receptors are really plentiful in smooth muscle fibers of the afferent arterioles. So at rest, sympathetic stimulation is going to be quite low. And this is when the afferent and efferent arterioles are going to be dilated. And this is when renal autoregulation of GFR is really going to be the main driving factor for glomerular filtration rate. Now, when there is moderate sympathetic stimulation, both the afferent and efferent arterioles constrict to the same degree. So moderate sympathetic stimulation. The afferent and the efferent arterioles constrict to the same degree. So this is when the blood flow into and out of the glomerulus is going to be restricted to the same extent. So this is going to decrease GFR just slightly though. With greater sympathetic stimulation though, with exercise or hemorrhaging, the vasoconstriction of the afferent arterioles is going to really dominate. So afferent arterioles are going to be very highly constricted, whereas the efferent arterioles are not going to be constricted as much. So as a result of this, blood flow into the glomerular capillaries is greatly decreased and GFR drops. Now, lowering renal blood flow kind of has two consequences. First one is that urine output is going to be decreased or reduced. This does help to conserve overall blood volume. But this also allows for greater blood flow to other body tissues. So these are consequences of lowering renal blood flow, not necessarily bad consequences, but just consequences of decreasing renal blood flow. Lastly, there is the hormonal regulation of GFR. So remember, there are two hormones that contribute to regulating GFR. We have angiotensin II, which decreases GFR, and atrial natriuretic peptide, which increases GFR. So first, angiotensin II, this is a very potent vasoconstrictor, and it narrows both the afferent and the efferent arterioles, and it reduces renal blood flow, thereby decreasing GFR. Then there's also going to be ANP. So cells in the atria of the heart secrete the atrial natriuretic peptide, and stretching of the atria that occurs when the blood volume increases. This stimulates the secretion of ANP. So here we have that ANP is secreted in response to stretching of the cardiac atria. Now, by causing relaxation of the glomerular mesangial cells, ANP increases the capillary surface area, and this is going to be the surface area that's available for filtration. And GFR is going to increase as surface area increases. So the more surface area available for filtration is going to allow the overall glomerular filtration rate to increase because of that. So you don't need to memorize this or anything, but this table shows us the comparison of the amounts of substances that are filtered, reabsorbed, and secreted. So here we have filtered in the second column here, reabsorbed in this third column, and then secreted in the last column. Now the volume of fluid entering the proximal convoluted tubules in just half an hour is greater than the total blood plasma volume because the normal rate of glomerular filtration is so high. So obviously some of this fluid has to be returned somehow to the bloodstream, otherwise we would have like no volume in our blood. So reabsorption is the second basic function of the nephron and the collecting duct. Remember, reabsorption meaning the return of most of the filtered water and many of the filtered solutes back to the bloodstream. So we're going to get that reabsorption both by active and passive processes. Now, once fluid passes through the proximal convoluted tubules, cells that are located more distally are going to fine-tune the reabsorption process to maintain the homeostatic balances of water and other selected ions or glucose, amino acids, selected molecules. Most small proteins and peptides that pass through the filter are also going to be reabsorbed, but this is usually done by pinocytosis. Then the third function of nephrons, remember, is going to be that tubular secretion. So this is going to be the transfer of materials from the blood and tubule cells into the glomerular filtrate. So as a result of tubular secretion, certain substances pass from the blood into the urine, and these kind of things can be detected by a urinalysis. So things like getting rid of toxic and foreign substances, these are things that can be detected by urinalysis. Fluid can leak between the cells of the renal tubule in a passive process, which is known as paracellular reabsorption. So even though the epithelial cells are connected by tight junctions, the tight junctions between cells in the proximal convoluted tubule are going to be pretty leaky, and they do allow some reabsorbed substances to pass between the cells into the peritubular capillaries. And then there's also transcellular reabsorption. So in this reabsorption route, a substance can pass from the fluid in the tubular lumen through the apical membrane of a tubule cell, across the entire cell through the cytosol, and then out into the interstitial fluid through the basolateral membrane. So here we have the couple different ways that substances can be reabsorbed. We have paracellular reabsorption up here, and then transcellular reabsorption here. Now, not many substances can be transported by paracellular reabsorption, but this is just the simple diffusion of substances between those leaky junctions between cells, and then just gets simply diffused into the peritubular capillary. So this is between those tubule cells in the renal tubules. The other way is by transcellular reabsorption. So for the example here for sodium, just like other cells throughout the body, the cells lining the renal tubules have a low concentration of sodium in their cytosol. And this is due to the action of sodium-potassium ATPases. Now these are only found in the basolateral membrane. You can see here. So this is a form of active transport. It requires ATP. And it allows the ejection of sodium into the interstitial fluid, which is then diffused into the peritubular capillary. Now these pumps are only found in the basolateral membrane, which really allows for a one-way movement or a one-way process of sodium. So because they're not found in the apical membrane, they're not going to be pumped out into the lumen of the tubule. Now recall that transportive materials across membranes can either be active or passive. Remember that primary active transport, the energy derived from the hydrolysis of ATP is used to essentially pump a substance across a membrane. So the sodium-potassium pump is a really good example of a primary active transport pump. In secondary active transport, this is where the energy stored in an ion's electrochemical gradient, rather than the hydrolysis of ATP, is going to drive another substance across a membrane. So secondary active transport really couples movement of an ion down its electrochemical gradient rather than the hydrolysis of ATP in order to drive another substance across a membrane. So it couples the movement of an ion down its electrochemical gradient to the uphill movement of a second substance against its electrochemical gradient. And then there's two types of secondary active transport. We have symporters. These are membrane proteins that move two or more substances in the same direction across a plasma membrane. And then antiporters will move two or more substances in opposite directions across a plasma membrane. We just need to remember that secondary active transport couples the movement of two separate materials. One is going to be down its electrochemical gradient, so it naturally wants to move that way, and it couples that and uses that to power the movement of another substance against its concentration gradient. Solute reabsorption drives water reabsorption. And this is because all water reabsorption occurs by osmosis. About 90% of the reabsorption of water filtered by the kidneys occurs along with the reabsorption of solutes like sodium, chloride, and glucose. So water that's reabsorbed with solutes in tubular fluid, this is termed obligatory water reabsorption. So again, this is about 90% of all water reabsorption. Now it's called obligatory water reabsorption because water is obliged to follow the solutes when they're reabsorbed. So they're like required to follow those solutes. This type of water reabsorption occurs in the proximal convoluted tubule, so it occurs in the PCT, and also the descending limb of the nephron loop. So descending limb of the nephron loop. And this is because these segments of the nephron are always permeable to water. So the proximal convoluted tubule and the descending limb of the loop of Henle are always going to be permeable to water. Now, reabsorption of the final 10% of the water is termed facultative water reabsorption. The term facultative, this means capable of adapting to a need. So this type of water reabsorption is regulated by the antidiuretic hormone, and this one occurs mainly in the collecting ducts. And we'll see why it's an adaptive type of water reabsorption in just the next slide. So like I said, the largest amount of solute and water reabsorption from filtered fluid occurs in the proximal convoluted tubule. And the proximal convoluted tubule, this reabsorbs about 65% of the filtered water, sodium, potassium, and then about 100% of most filtered organic solutes like glucose, amino acids, and then a bunch of other ions. Most solute reabsorption in the proximal convoluted tubule, this is going to involve sodium. And sodium transport occurs by symport and antiport mechanisms in the proximal convoluted tubule. So a main one is going to be the sodium glucose symporters. These are found in the apical membrane of a cell in the proximal convoluted tubule. In another secondary active transport process, the sodium hydrogen antiporters, they will carry filtered sodium down its concentration gradient into a proximal convoluted tubule cell. And during that process, hydrogen is moved from the cytosol into the lumen. Now, cells lining the PCT and the descending limb of the nephron loop are especially permeable to water because they have many molecules of aquaporin 1. Aquaporin 1 is a membrane protein that allows the membrane to be permeable to water. Kind of makes these little holes in the membrane that water can pass through. We will see a little bit later that another type of aquaporin is used in the collecting ducts, and the antidiuretic hormone actually affects how many aquaporin 2 molecules are inserted in the collecting ducts, but we will see that a little bit later. But aquaporin allows water to pass through membranes. Okay, so here we have some symporters and antiporters in the proximal convoluted tubule. So first one we'll look at is the sodium glucose symporter, this figure on the left here. Again, this is found in the apical membrane. I'll use a different color rather, but the apical membrane of the cells in the proximal convoluted tubules. So you can see it right here. So this uses sodium and a molecule of glucose. So sodium and a molecule of glucose will each attach to this importer protein, which is going to carry them from the tubular fluid into the tubule cell. The glucose molecules will then exit by the basolateral membrane by facilitated diffusion transporter. So we can see that one here. And then they're going to just simply diffuse into the peritubular capillaries here. And then sodium, we've already seen how sodium will move by the sodium-potassium pump and then diffuse also into the capillary. The next figure we have here on the top right is going to use the sodium-hydrogen antiporters. You can also see this in the apical membrane of the PCT cell. So they're going to carry sodium down its concentration gradient into the tubule cell. Because remember, the concentration of sodium within cells is going to be very low compared to outside of cells. So it's going to be down its concentration gradient. And at the same time, hydrogen is going to be moved from the cytosol into the lumen. Sodium will be reabsorbed into the blood, and hydrogen is secreted into the tubular fluid to be secreted or excreted rather. The PCT cells will continue to produce the hydrogen needed to keep the antiporters running in this way. Then we have the last figure on the bottom right, shows reabsorption of HCO3. You can see this here. Now, most HCO3 in the filtered fluid is reabsorbed in proximal convoluted tubules. And really this safeguards the body's supply of a really important buffer. So after hydrogen is secreted into the fluid within the lumen of the proximal convoluted tubule, it reacts with filtered HCO3 or bicarbonate, and it's going to form carbonic acid, H2CO3. And carbonic acid is going to really readily dissociate into water and CO2. The carbon dioxide is then going to diffuse back into the tubule cells and rejoin with water to produce carbonic acid. And carbonic acid is then going to dissociate into a hydrogen ion and bicarbonate within the cell. Now, as the levels of bicarbonate, HCO3, rise within the cytosol, it's going to exit by facilitated diffusion transporter. You can see this right here. And then it's going to diffuse into the blood along with sodium. So for every hydrogen ion that's secreted into the tubular fluid of the proximal convoluted tubule, 1 bicarbonate and 1 sodium are reabsorbed. Now, solute reabsorption in the proximal convoluted tubule really promotes the osmosis of water. So again, this is still in the proximal convoluted tubule. Each reabsorbed solute increases the osmolarity, first in the tubule cell, and then in the interstitial fluid, and then finally in the blood. So water is going to move quite rapidly from the tubular fluid by both paracellular, so remember paracellular is here, and also by transcellular routes. So water is really just going to be rushing to follow these solutes that are being reabsorbed, where water is really just working to restore osmotic balance. So in other words, reabsorption of the solutes, like these solutes here, this is going to create an osmotic gradient that promotes the reabsorption of water by osmosis. Cells lining the proximal convoluted tubule and also the descending limb of the nephron loop are really permeable to water. Remember, this is because they have many molecules of the aquaporin 1. Now, that is aquaporin 1 is an integral protein in the plasma membrane, and essentially it's just a water channel that greatly increases the rate of water movement across both the apical and basolateral membranes. Now, because all of the proximal convoluted tubules reabsorb about 65% of the filtered water, fluid is going to enter the next part of the nephron. The nephron loop, or the loop of Henle, is what we're going to talk about next. This is where, for the first time, reabsorption of water by osmosis is not automatically coupled to the reabsorption of filtered solutes. And that's because this part of the nephron loop is not really permeable to water. It's relatively impermeable to water, really, especially in the ascending limb. So the most permeable to water is going to be that proximal convoluted tubule. In the descending limb of the loop of Henle, it's a little bit permeable to water, but once we get to the ascending limb of the loop of Henle, relatively impermeable to water. Water cannot follow the solutes that are reabsorbed. So the nephron loop really sets the stage for this independent regulation of both the volume and the osmolarity of body fluids. Now, the apical membrane of cells in the thick ascending limb of the nephron loop have sodium, potassium, chloride symporters. So here we can see this apical membrane. Again, it's going to be really impermeable to water in that thick ascending limb of the loop of Henle. And we can see these, actually I'll just highlight this here, these sodium, potassium, chloride symporters. We can see it here. Now these transporters will simultaneously reclaim 1 sodium, 1 potassium, and 2 chloride from the fluid in the tubule lumen. Now we already know sodium that's actively transported into the interstitial fluid at the base and sides of the cell are going to just diffuse very quickly into the vasa recta here. Chloride is going to move through these leakage channels at the basolateral membrane. It's going to diffuse into the interstitial fluid and then diffuse directly into the vasa recta. Now, there are many of these potassium leakage channels in the apical membrane. So because of this, most of the potassium that's brought into the cell is going to move down its concentration gradient and kind of just travel right back into the tubular fluid. So because of this, the main effect of the sodium, potassium, chloride symporters is really just going to be the reabsorption of sodium and chloride. We're now going to move on to talking about the distal convoluted tubule, or DCT. The early or initial part of the distal convoluted tubule reabsorbs about 10 to 15% of filtered water, 5% of the filtered sodium, and about 5% of filtered chloride. Now, reabsorption of sodium and chloride occurs by means of the sodium chloride symporters. And these are found in the apical membranes. Sodium potassium pumps and chloride leakage channels in the basolateral membranes will then allow for reabsorption of sodium and chloride into the peritubular capillaries. The early DCT is also a major site where the parathyroid hormone, or PTH, stimulates the reabsorption of calcium ions. Now, the amount of calcium reabsorption in the early DCT really varies depending on the body's needs. By the time fluid reaches the end of the distal convoluted tubule, about 90 to 95% of the filtered solutes and waters have been returned to the bloodstream. Now recall that two different types of cells, principal cells and intercalated cells, are present at the late or terminal part of the distal convoluted tubule, and they're also found throughout the collecting duct. The principal cells here at the top, these reabsorb sodium and secrete potassium. These cells are also stimulated by antidiuretic hormone in order to insert more aquaporin II molecules into their membranes. Intercalated cells, these reabsorb potassium and a bicarbonate, and they secrete hydrogen ions. A negative feedback system involving antidiuretic hormone regulates the facultative water reabsorption. With the osmolarity or osmotic pressure of plasma and interstitial fluid increasing, that is when water concentration decreases by as little as 1%. This is when those osmoreceptors in the hypothalamus are going to detect that change. So here we have an increasing osmolarity of the plasma and interstitial fluid. It's detected by osmoreceptors in the hypothalamus. Their nerve impulses are going to stimulate the secretion of more antidiuretic hormone into the blood. So we're going to increase ADH in the blood, and the principal cells become more permeable to water. So this is going to increase facultative water reabsorption, and as that facultative water reabsorption increases, the plasma osmolarity decreases back to normal, bringing us back to homeostasis. So here we have a nice negative feedback loop. Even though your fluid intake throughout the day or from day to day can be really highly variable, the total volume of fluid in your body normally remains quite stable. Homeostasis of body fluid volume depends in large part on the ability of the kidneys to regulate the rate of water loss in urine. Normal functioning kidneys produce large volumes of dilute urine when fluid intake is high, and they can also produce a small volume of concentrated urine when fluid intake is low or fluid loss is high, say you're excessively sweating. Antidiuretic hormone is going to control whether dilute urine or concentrated urine is formed. So in the absence of ADH, urine is very dilute. So it's going to be very high volume, very light in color, so it contains a lot of water. However, a high level of antidiuretic hormone is going to stimulate the reabsorption of more water into the blood, creating a more concentrated urine. So it's going to have very low water content, very high solute content. So usually it's going to be lower volume. Usually more darker yellow in color, whereas dilute urine is going to be very light in color. So again, when you have a high intake of fluids, your kidneys will produce dilute urine in high volumes. And with a low fluid intake, your kidneys produce very concentrated urine, but in a low volume. Glomerular filtrate has the same ratio of water and solute particles as blood does. Its osmolarity is about 300 milliosmoles per liter. So fluid leaving the proximal convoluted tubule is still isotonic to blood plasma. The ability of antidiuretic hormone to cause excretion of concentrated urine depends on the presence of an osmotic gradient. So if there's an osmotic gradient of solutes in the interstitial fluid of the renal medulla, antidiuretic hormone can cause the excretion of concentrated urine. When dilute urine is being formed, the osmolarity of the fluid in the tubular lumen increases as it flows down the descending limb, and it decreases as it flows up the ascending limb of the nephron loop, and then it decreases even more as it flows through the rest of the nephron and the collecting duct. So this figure here shows the formation of dilute urine. So we can see the osmolarity of interstitial fluid in the renal cortex being 300 milliosmoles per liter. And you can also see that the interstitial fluid in the renal medulla gets more concentrated the further down you go into the medulla. So you can see here we're starting at 350 and we get more concentrated as we go deeper. And because of this, more and more water is reabsorbed by osmosis as the tubular fluid flows along the descending limb toward the tip of the nephron loop down here. So here we're going to get more and more water reabsorbed. So water is going to be leaving the loop here and being reabsorbed into the blood as we go lower and lower. And this reabsorption of water is going to cause the osmolarity of the fluid within the tubule to increase until we get to its maximum down here at the tip of the loop of Henle. So you can see that the osmolarity, or the concentration of solutes within the fluid, is going to increase in the descending limb till we get to the end of the loop of Henle. And then as we go up in the ascending limb, osmolarity is going to decrease. And once we get to the distal convoluted tubule here, it's quite low already because more solutes are continuing to leave. They're continuing to be reabsorbed. So solutes are being reabsorbed here. but very few water molecules are being reabsorbed at this point. So lots of solutes leaving, but a lot of the water is staying within the tubule at this point in the nephron. Remember those early distal convoluted tubules cells, they're not very permeable to water, and they're not regulated by ADH. And then when we get into the collecting duct here, osmolarity continues to drop until we get very dilute urine here. So continuing to decrease in osmolarity as we go down the collecting duct. Cells lining the thick ascending limb of the loop of Henle have symporters that are actively reabsorbing sodium, potassium, and chloride from the tubular fluid. So the ions will pass from the tubular fluid into the thick ascending limb cells, then into the interstitial fluid, and then finally will diffuse into the blood inside the vasa recta. Now, even though these solutes are being reabsorbed in the thick ascending limb, the water permeability in this portion of the nephron is really quite low. So water cannot follow these ions by osmosis. As the solutes, but not water molecules, are leaving the tubular fluid, its osmolarity drops to about 150 milliosmoles per liter. And we saw that in the last picture. And you can refer back to that if you need. So the fluid entering the distal convoluted tubule is going to be more dilute than blood plasma at this point. The fluid continues flowing along the distal convoluted tubule. At this point, additional solutes will be reabsorbed, but very few water molecules. And the early distal convoluted tubule cells are not very permeable to water, and they're also not regulated by antidiuretic hormone. Finally, we get to the principal cells of the late distal convoluted tubule and also the collecting duct. And these cells are going to be impermeable to water when antidiuretic hormone level is low. So very low water permeability in absence of antidiuretic hormone. So in this case, the tubular fluid becomes progressively more dilute as it flows onward. And this is because more solutes are continuing to leave, continuing to be reabsorbed, but water cannot cross the barrier and leave the tubular fluid to go into the blood. So the water all has to stay, but the solutes are being removed. So by the time that the tubular fluid drains into the renal pelvis, its concentration can be as low as 65 to 70 milliosmoles per liter, and that is like 4 times more dilute than blood plasma or glomerular filtrate. So that was how dilute urine is formed. So let's look at how concentrated urine is formed. So when water intake is very low or water loss is high, for example, during heavy sweating, the kidneys must conserve water while still being able to eliminate wastes and excess ions. So this is where we're going to talk about these really long looped juxtamedullary nephrons, which are more used for concentrated urine. So the ability of the antidiuretic hormone to cause excretion of concentrated urine, like I said, depends on that osmotic gradient, briefly mentioned before. So this is an osmotic gradient of solutes in the interstitial fluid of the renal medulla. Now there's going to be 2 main factors that contribute to building and maintaining this osmotic gradient. First off, these are going to be the differences in solute and water permeability and reabsorption in different sections of the nephron loops and collecting ducts. So, solute and water So solute and water permeability and reabsorption differences in different nephron sections. So this means is that the water the permeability of water and the reabsorption of water and solutes is going to be different in different areas. The second one is called countercurrent flow. So this is going to be the countercurrent flow of fluid through tube-shaped structures in the medulla. So what that just means is that the flow of tubular fluid in the nephron is going to be going one direction, where the flow of blood within the blood vessels is going to be going a different direction. And they're going to be flowing in these different directions right next to each other. So countercurrent flow just refers to the flow of fluid in opposite directions. So this diagram here, we have the formation of concentrated urine. So at the end here, we can see concentrated urine, and we're going to go over how it is created. So first off, we're going to start over here at number 1. This is where the symporters in the thick ascending limb cells of the nephron loop cause a buildup of sodium and chloride in the renal medulla. So this is where we have those sodium, potassium, chloride symporters. So it's going to be by those green arrows we can see here. So this is where we get those specific symporters that reabsorb sodium and chloride from the tubular fluid. Remember, the potassium just kind of goes out by leak channels, so it's not as important with this symporter. The sodium and chloride are going to be more important with this symporter. At this point, the water is not reabsorbed in this segment, this thick ascending limb, and this is because the cells here are going to be impermeable to water. So pretty much up to this point, it's going to be very similar to the last slide where we saw the formation of dilute urine. We have this osmotic gradient, but because the loop of Henle is so much longer, it goes a lot deeper into that very strong osmotic gradient. Whereas in the last one, we're only getting to about 900 milliosmoles. So the other cortical nephron loops are only going to be coming down to about here. So not that far into the osmotic gradient of the medulla. Whereas these really long juxtamedullary nephron loops are going to go much deeper into that osmotic gradient. So we're going to get a much higher osmolarity at the bottom of that loop of Henle. But essentially it's going to be the same. We're going to have 300 milliosmoles at the end of that proximal convoluted tubule. As we go down the descending loop, or descending limb of the loop, water is going to be leaving just like we saw before, which means that inside the tubule, that concentration is going to be increasing. And then we get to the symporters in the thick ascending limb. We get that buildup of sodium chloride. Number 2 covers the countercurrent flow. So this is going to be countercurrent flow through the descending and ascending limbs of the nephron loop, and this helps to establish an osmotic gradient in the renal medulla. So since that tubular fluid constantly moves from the descending limb to the thick ascending limb of the nephron loop, that thick ascending limb is going to be constantly reabsorbing sodium and chloride, and the sodium and chloride that is reabsorbed here contributes to the osmotic gradient overall in the interstitial fluid. Next, we have number 3 over here, which covers the principal cells in the collecting duct. Now these are going to be reabsorbing a lot more water when the antidiuretic hormone is present. So if you are losing a lot of water through sweat, or you're drinking or consuming a lot less water throughout the day, antidiuretic hormone is going to be high, and that's going to cause those principal cells in the collecting duct to increase their permeability to water, and water is going to very quickly then move out by osmosis into the interstitial fluid and then into the vasa recta. So we're going to have water leaving, which is going to be increasing that osmolarity as we go down into the collecting duct. Lastly here, number 4 is going to involve urea, and this is going to be urea recycling. So this urea recycling causes a buildup of urea in the renal medulla. So urea leaves the collecting duct in this area, becomes accumulated in the interstitial fluid, and some of it will diffuse into the tubular fluid, into the descending and thin ascending limbs of the long nephron loops, which are going to be permeable to urea. But this just also adds to the osmotic gradient. So that by the end, we have very concentrated urine here at about 1200 milliosmoles per liter. As fluid flows along the collecting ducts, water reabsorption continues by osmosis, and this is because antidiuretic hormone is present. Antidiuretic hormone causes collecting ducts to become very permeable to water. So water reabsorption is going to further increase the concentration of urea in the tubular fluid, and then more urea diffuses into the interstitial fluid, and then the cycle is going to repeat. So we have that movement of urea contributing to osmolarity in the interstitial fluid in the renal medulla. Here is a really nice summary slide going over the different sections of a nephron and what is filtered, reabsorbed, and secreted in each section. So here we have the proximal convoluted tubule. The main things are the reabsorption of water, sodium, potassium, glucose, and amino acids. This is going to be the main thing that happens at the PCT, is going to be reabsorption of a lot of things. Then we have the loop of Henle down here, reabsorbing just a little bit more of that water and sodium chloride and potassium as well. Then in the distal convoluted tubule, again, just a little bit of reabsorption of water with very minimal reabsorption of sodium chloride and calcium. But remember that calcium here is going to be reabsorbed, stimulated by that parathyroid hormone. And this is going to be the main area where that calcium is going to be reabsorbed. Then we can also see the principal cells in the late distal convoluted tubule and the collecting duct. Going to be reabsorbing more of that water, again stimulated by antidiuretic hormone, and that urea recycling to the loop of Henle. So those are going to be important. And then a little bit with the intercalated cells here. Just a little bit with that bicarbonate reabsorption and hydrogen secretion. So again, just a review of everything that we've kind of gone over so far. Routine assessment of kidney function is going to involve evaluating both the quantity and quality of urine and the levels of wastes in the urine and in the blood. So it's important to check the presence of abnormalities of things like albumin, which is normal in blood plasma in small amounts. glucose, which is normally not in urine at all. Red blood cells, again, normally not in urine. Ketone bodies are sometimes present in really small quantities, so if you find small quantities, that could be normal. And then also microbes in the urine as well. Usually when they do a urinalysis, they're going to, if there are microbes present, they're going to test for the quantity and type of bacteria, and the type of bacteria will vary with specific types of urinary tract infections. So just a bit on transportation and storage of urine. From the collecting ducts, the urine drains into the minor calyces, which join to become major calyces that unite to form the renal pelvis. From the renal pelvis, urine first drains into the ureters and then into the urinary bladder. Urine is then discharged from the body through a single urethra. Each of the two ureters transports urine from a renal pelvis of one kidney to the urinary bladder, and this is done by peristaltic waves or peristaltic contractions of the muscular walls of the ureters, and also by hydrostatic pressure and gravity also contribute. Now there's no valve at the opening of the ureters into the bladder, but when the bladder fills, it compresses the openings near the top and it prevents backflow. A hollow bladder has a capacity of about 700 to 800 milliliters. At the base of the urinary bladder, the ureters curve medially and pass obliquely through the wall of the posterior aspect of the urinary bladder. So we can see these ureteral openings. The ureters come in kind of from the backside and have openings here. The urinary bladder is a hollow, distensible muscular organ that is situated in the pelvic cavity just posterior to the pubic symphysis. In the floor of the urinary bladder is this small triangular area. It's here in these dashed lines, and this is called the trigone. Trigone just means triangle. So the posterior two corners of the trigone contain the two ureteral openings. And then the opening to the urethra, so here's the urethra, but the opening to the urethra is the internal urethral orifice. This makes up the anterior corner. Now, because the mucosa is firmly bound to the muscularis in this area, the trigone has quite a smooth appearance. You can also see rugae in the rest of the bladder. These are going to be folds in the mucosal layer, and this allows for expansion of the urinary bladder. Around the opening to the urethra, there are circular muscular fibers that create or form an internal urethral sphincter. And just inferior to it is the external urethral sphincter. And the external urethral sphincter is composed of skeletal muscle, and it's actually a modification of the deep muscles of the perineum. The internal urethral sphincter is involuntary, whereas the external urethral sphincter is voluntary, and it controls the opening and closing of the urethra. Discharge of urine from the urinary bladder is called micturition. This is also known just as urination or voiding. Micturition occurs by a combination of involuntary and voluntary muscular contractions. When the urinary bladder volume exceeds 200 to 400 milliliters, the pressure within the bladder increases considerably, and stretch receptors within the bladder or in the wall of the bladder will transmit nerve impulses to the spinal cord. Urinary bladder filling causes a sensation of fullness that initiates a conscious desire to urinate before micturition reflex actually occurs. Even though emptying of the urinary bladder is a reflex, in early childhood we actually learn to initiate and stop it voluntarily. Like I said, the urinary bladder capacity averages about 700 to 800 milliliters. It's smaller in females because the uterus actually occupies the space just superior to the urinary bladder. In males, the urinary bladder is directly anterior to the rectum. And in females, it's anterior to the vagina and inferior to the uterus. Now, the urethra is a small tube leading from the internal urethral orifice in the floor of the urinary bladder to the exterior of the body. In both males and females, the urethra is the terminal portion of the urinary system and the passageway for discharging urine from the body. In males, it discharges semen as well. The male urethra, which also consists of a deep mucosa and superficial muscularis, is divided into three anatomical regions. The prostatic urethra, which passes through the prostate. The intermediate or membranous urethra is the shortest portion. It passes through the deep muscles of the perineum. And the spongy urethra, which is the longest portion, passes through the penis. In females, the urethra lies directly posterior to the pubic symphysis. It's directed obliquely, inferiorly, and anteriorly, and it has a length about 4 centimeters. The opening of the urethra to the exterior is by the external urethral orifice, both in males and females. There are a couple differences between the male and female urethras. It's 5 times longer in males. It's divided into 3 segments in males, but only a very short tube in females. And it is a common duct for urinary and reproductive systems in males, whereas in females, the two systems are very distinctly separate. And that will be the end of chapter 25.